Okay, so feel free, you can ask questions, that's all fine. Okay, this is our slideshow on microscopes. Come on, kind of bad if we don't have an internet connection. Okay, and you can see a lovely little picture there with the compound eye of flies. Each of those is a lens in its own right. So whereas you have one lens on the back of your eye, it has hundreds. Okay, it can lose. It can have, dozens of those destroyed. It can still see fine. Whereas of course if you take a hit to the eye, you're in a fair amount of trouble. Okay, it's necessary for a small insect. Key learning intentions. What is the difference between light microscopes and electron microscopes? There's some, you won't get to use an electron microscope. The university has them, but we do not. Teach students how to use a light microscope. We've done some work with that already. Teach students the rules of scientific drawing. You're already pretty familiar with those based upon our podcast. Okay, and there are some key terms that you need to be familiar with. And that's a little picture of dimetons. It's not somebody's tile in their, from their bathroom floor or something like that. It's just neatly arranged little animals that you can find in water. And you can see there's odd little shapes like triangles and things like that. Okay, they're kind of cool. Okay. You don't need to write those down because you've got them on the key learning intention document. Now we saw this slide before, remember? A long time ago for you guys. 6 weeks, give or take. We talked about, you'll see this more in Year 9 in disease, but in the Middle Ages, half the children died before the age of 15. So you'd have 4 children, well, you'd probably have 8 children, but 4 would die before the age of 15. Or if the Black Death came through, maybe all of them. Okay, maybe the entire family was killed. Okay. Disease was caused by humours and evil spirits and curses and gods' angers, different gods. Yep.
What does it mean by the 4 humours?
Earth, fire, air and water. And so you've got bile and you've got, I can't even remember what they are, choler, bile and things like that. And they just had different ideas. None of them were right. There's no scientific basis to them. There was no scientific method. The idea that I have an idea, I come up with a hypothesis, I test it with a control variable, an independent variable, and then I share my results with other people who then do the same work. And I write it all down. It's really important. None of that happened. So, people just guessed. Leprosy destroys nerves, blood vessels, ligaments, tissues, erodes bones. There's a picture of a leper there. What's the bell for?
To warn away people.
Yes, ring the bell to warn people away. Don't come near me. I'm a leper. And they would survive by begging. Okay, and if you look in the Bible, but any religious document really, people with leprosy were considered like they'd been cursed in some terrible way because it destroys everything, and you look like you're rotting. Okay, it's caused by a bacteria. If you get leprosy now, they can just give you antibiotics. A lot of them, sure, it's a tough bacteria, but they can kill it. Okay, but microbes were unknown at that time. No microscopes. Medieval medicine. Physicians and apothecaries used mathematics, astronomy, religion to treat patients. Bloodletting by barbers was used to remove bad blood. That's apparently how Robin Hood died. They just cut the wrist and were letting the bad blood out. If you overbleed people, they die. Also, of course, if you're really sick, if I have a bacterial infection, maybe I've cut myself and I have a bacterial infection, so I'm very, very sick, and then you remove 2 litres of my blood, that's not going to help. It's going to make me feel a lot worse, okay? You know the barbershop poles?
Yeah.
Why do you think they're red and white?
Blood and bandages.
Blood and bandages. Barbers have been running around for hundreds of years. I'll cut your hair, I'll pull teeth, I'll let blood out if you're feeling sick. So yes, blood and bandages on the barbershop pole. That's how long ago that was. You've seen this before. This idea of miasma theory, bad air. It's not a crazy theory. I'm coughing, and then Jamie starts coughing a day later, and they're going, there's something going on there. But they actually thought my infection was caused by bad air. Okay, this is blue, so you don't have to write this down. The summary is a little bit more important, so you should write that down. Okay. The idea was that Black Death was caused by bad air, but it's not. It's just a bacteria again. And we looked at the plague doctor's costume from before. And not a bad idea, actually. Not to actually have a robe over the top of your clothes, so that if people spit blood or mucus on you, or they burst in some other revolting way, it doesn't get on you. Yes? The hat, yeah, keeps everything together. Even the mask is not a terrible idea. Okay, they stuffed sweet-smelling things in there to keep the bad air away, but it actually would filter out bacteria to a certain extent as well. So not the worst idea in the world, but the whole idea of bad air was erroneous. However, summary, pre-science people believed diseases were caused by the environment. They got that right. They knew that if a tree fell on you, you'd feel sick, you know, or maybe if you ate bad food, you'd feel sick. They knew that maybe more recently, obviously, a good example I like to use always is coal dust. If you breathe a lot of coal dust, you're going to get black lung. You know, you're going to cough and cough and cough. It destroys your lungs. So they had this idea that things in the environment could get you. They were on top of that. What they didn't understand was, you know, they used imbalanced humours and witchcraft and sorcery and God's curses and bad air as causes for disease, and none of that's true. They didn't know about pathogens. They didn't know about genetics. Pathogens, with microscopes, that started to come through. So that's why microscopes were really important, and genetics not until considerably later on, only in the last 100 and a bit years. 1953, DNA was demonstrated to be the molecule of inheritance. That's not long ago. Your grandparents would remember that, maybe, if they were paying attention, if they weren't on their phones obsessively texting. That's a trap. Mobile phones weren't invented until considerably later. Can I change?
No. One second.
Okay. I try to keep the word count down. Okay. These key points: environment, pathogens, genetics. There's 3 ways to get sick. Yeah, and that's it. You know, and you will need to remember those. Not for this course actually, but for year 9 certainly. And one of the reasons that microscopes are important is for pathogens. And that allowed doctors to examine things and say, hang on a minute, there's a whole world down there, you know. And then, if you're cluey, you start to ask questions like, this person over here is going all rotty and mouldy for some reason, and I'm going to take some samples and have a look at them. And do I see anything there that I'm not familiar with? Could that perhaps be causing the disease? The rotty mouldiness, or whatever it might be. Okay. Right. There's an early microscope. You don't need to know about Hooke. He's one of the pioneers of microscopy. That's a picture from his book. Okay. And he introduced the term cell into the vernacular. That's one of the important things he did. Okay. He introduced the term cell because the walls of dead cork cells reminded him of small rooms that monks lived in. Okay. And so this idea of cells, which is our next unit of work, is from Hooke. Okay, and I think this is cork, if I'm not mistaken. So he was just looking at cork, and he saw these regular structures. Tung, 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 tung. And then when he started looking at other things, he saw the regular structures, and he goes, oh, that's interesting, and used the word. Okay, that's an early microscope. It kind of looks like a telescope in its own right. And there's an oil lamp and some lenses to provide light to the sample, because you've seen that yourself, that when you're looking at something very small, it can look very dim unless you have a bright light source nearby. Right, I should be able to change. Okay, Van Leeuwenhoek, you do need to know about. Used a simple microscope and saw tiny shapes in pond water. Okay, and I think we talked about these before. I've shown you that slide before. Because they were moving, he decided they were alive. Is that a valid assumption? Movement equals life. When we talk about living things, what are we really interested in? What's our definition for living things? Yes? If you're thinking Mrs. Gren, yeah, which we did before, you need to be conscious of Mrs. Gren. But of course he was right. They're alive. And then people started doing cool little things. And did I show you the video on hydras?
Yes.
They're cool. These ones here,
One that could never.
yeah, they're immortal as far as we can tell. You cut them in half, they just get better. And as long as something doesn't kill them, they'll keep going. If they can be eaten and digested, sure, that will kill them. They can be squashed easily. They can dehydrate. But if you put them in an ideal environment and leave them alone, they'll just keep replicating and doing their thing quite happily. And we'll check out that little video on dimetons in a little bit. I won't do it now because that will waste video time. But there's a video on dimetons, the sorts of things that you see in water. And, you know, they're happy little things. Okay. Yes. First microbiologist to study muscle fibres, bacteria, spermata, sperm, and blood flow in capillaries. So he's a busy man. Okay, so you can see straight away that with these early microscopes, people just started looking at things, and it started to dramatically change the way people thought about the world and saw the world. What's that weird theory that was completely wrong? It's spontaneous generation, I think it's called. See, people at this time would think, oh look, okay, bad food turns into mice, okay? Because what you do is you're like, I've got some grain and I'm going to put it in a drawer over there, and if I come back 2 weeks later and open the drawer, there'll be mice in there. So the grain has turned into mice. It's completely fallacious, it's illogical. There are all sorts of strange things. What is disease caused by? Oh, you know, angry gods and this sort of thing. And it's not until the scientific method really came through that people would start to conduct experiments. And you look at next year at Louis Pasteur and the work that he did, and Robert Koch and the work that he did on establishing, you know, which particular bacteria caused disease. And Pasteur did some critical work on demonstrating that when things go bad, it's because of bacteria. It has nothing at all to do with anything else. Now, this is the compound microscope. This C up here means that's effectively a cram card. If you're an inherently lazy person, you could just print that off. Print it off, stick it in your study notes, and off you go. You don't need to photo it. You can if you like. It's not an issue, but it's online and it'll print off nicely. But that's what the C stands for. So I'll try and keep giving you those hints, okay? I've got other hints here, like the red dots are important, because what's the difference between those two? A compound light microscope, you need to know what that is, uses focused light and lenses to magnify a specimen, usually a cell or cells. An ocular lens, this one over here, okay, has maybe a magnification of 10 times. They can go higher, but it's usually 10 times. The objective lenses, you've seen those are the ones you rotate around. There's your objectives down there. Can be 4, 10, 40 and 100 times. They can go higher, but the microscopes here use 4, 10, 40, 100. You need to take notes. If it's red, it's important. Yeah? So, light wavelengths, you don't need to know this per se, are about this long. Okay, a nanometer is a billionth of a metre, so it's a millionth of a millimetre. So 380 millionths of a millimetre is how long a wavelength of light is. Okay, they're short, high energy short. Okay, so they allow magnification of about up to 2,000 times. Our microscopes only go to 1,000 times, right? How do I work out magnification? We'll look at this later, but it's this lens here, the top one, times whatever one you're looking through down the bottom. So that's the biggest one I have available to me. 10 times 100 is 1,000 times magnification. You'd have to have a very expensive, good quality light microscope to get out to 2,000. I think you'd be very lucky to find one that would even get you to 1,500. But because of the size of light wavelengths, that's the best you can do. Okay, you can't see anything smaller than that wavelength. You just can't do it. Okay, you can work on lenses all day. You cannot see anything that's smaller than the wavelength of light. Yes.
Do we need that bottom part?
No, not really.
What
I
about
think you understand that this is just some general knowledge. Like it's for magnifying things close up. small things close up. Like, don't write down, you don't have to write down everything. Just write down what you don't know or maybe struggle to understand.
What about that one?
No, you won't need that, but that's a part of background. This is the difference between electron microscopes and light microscopes. Electron wavelengths are much smaller, so you can see smaller things. An electron microscope has a magnification of 50,000 times, so 50 times greater than the ones that you're using in class. So you can get really, really good, clear pictures of very, very small things. And it's because of this difference in wavelength. So that is an important point.